Okay, so back to topic 8. I want to do a nice flow chart for you, so I'm going to start drawing that in a moment here. So if you have a piece of paper, you might want to get that handy. Sorry, I'm just going to plug this in. Okay, so this is where we left off, right? And I told you I want to make a flow chart and kind of talk a little bit about these organisms in particular. There are obviously a lot more organisms. Some of these are very easy to distinguish through just basic gram stain, maybe another test or two. And that's kind of what I want to distinguish doing a flow chart for you. So let's give it a try. See if I can draw. So where is it? There it is. Okay, so that's what my flow chart is going to look like. I may actually go to a second page just with the size of my drawing. My goal was to have gram negatives over here, or sorry, gram positives over here, gram negatives over here, but probably by the time I'm drawing all the gram positives, it's going to take all this space just in terms of writing on the computer screen. So I'll probably do a second page for the gram negatives, but this was the plan, right? To do this on paper using the webcam originally. So you can see the first step is collecting the sample, right? And then it's a matter of, you can do a gram stain or McConkey agar or other selective media to select for gram negatives or gram positives, right? And what I'll do is I'll draw some little pictures of the organisms too as we go along. So gram positives, we're looking at basically 3 scenarios. So let's see here, I need 10. Here we go. So first scenario is Streptococci. Second scenario is Staphylococci. And third scenario is Bacillus. All right, so we'll draw some little pictures just so we remember what these guys look like. So Streptococci, so that's purple, by the way, apparently. So there we go. So strepto is chain, cocci is spheres, of course. Staphylococci, they're clusters. And Bacillus are rod-shaped. There we go, some beautiful cells, right? Okay, so Streptococci and Staphylococci, the problem is, depending on your gram stain and whatnot, you may not see them in chains or clusters. If they're particularly diffuse, you know, you're only seeing a few scattered cells, they can often look exactly identical. So remember, we can do other tests, right? We have our Catalase test. So Streptococci are Catalase negative. So remember, that means no bubbles. I guess I could write that in there. I think there's enough room. And Staphylococci are Catalase positive. Okay, and then what we're going to do with the Streptococci is you can put that on blood agar. And remember, we had the alpha hemolytic. Sorry, let me just alpha hemolytic. Let's see if I can. How did I do this before? Oh, there we go. some more space for myself. Alpha hemolytic. Oh no, what did I do? Okay. Beta hemolytic and gamma hemolytic. So I'm not going to expand that part any further. That's what we did last day. So remember the alpha hemolytic is Streptococcus pneumonia. Maybe I'll write that one down because that's one of the important organisms for this class. So we had Streptococcus pneumonia. And beta hemolytic, we had Streptococcus pyogenes. Streptococcus gamma hemolytic, we had the Enterococcus, and there are other organisms, and like I said, I'm not going to go into all of them. These are the two you need to know for this class, so I'm just making sure that they're on this flow chart. If you want to see the full flow chart, this was last day's lecture, so you can go back and refer to that. Okay, so the Staphylococcus. Gonna have to expand over here somehow. And like I said, I kind of ended up being a lot messier when I'm doing this on the screen as opposed to on a piece of paper. But we can do that. So how did I move the screen a little bit? Move just a tiny bit. Maybe shrink it just a little bit. There we go. All right, Staphylococci. Okay, so here we go, over here. And on our way, we can do what is called a Coagulase Test. So I mentioned that one last day. The Coagulase Test is where it shows the clumping. So Coagulase Test, I'll write that one in there. So if you have clumping, we get Staphylococcus aureus, okay? So I'm not going to write a full name. And if you don't have clumping, you have Staphylococcus epidermidis. No. Clumping. And one more thing that's important to mention, and we were talking about a little bit, we're going to be talking about a lot in the next couple of weeks, is Staphylococcus aureus is a really very important hospital-acquired infection in many cases, and also a superbug, and goes by a lot of names. And so sometimes we need to know how serious it is. So one of the other tests that might be done is some antibiotic susceptibility. Often, initially, somebody might have a staph infection. They'll usually prescribe them some sort of routine antibiotic right away. Doesn't clear up start to clear up relatively quickly. They're going to start to do those tests. Or if there's reason to suspect that it is drug resistant, they might do an antibiotic susceptibility test. We're going to talk about how those things are done in topic 10. So we'll get there. So it's worth it to put that in there as a note. So here we go. Antibiotic come on, where's the one? Here we go. I don't have the most amazing handwriting, but it just pains me to see how much it's butchered by writing on the screen. As I joke with some of my other students, I just know my handwriting is way better than our chemistry instructors, so at least I have that going for me. Soren's handwriting is just awful. Okay, so there's one more gram-positive, which is the Bacillus, right? And there's a few species that are relevant for that. And I'm going to write them over here on the right? So we'll go over here. Unfortunately, there are many rod-shaped bacillus, okay? And that includes Bacillus species, okay? And I'm going to write a little note beside these each in a second here. I'm going to move the screen over, but I'm just going to write the names of the species first. What else do we had? We had Clostridium. So I'm just going to say, by the way, if you don't get all this, or if it's just a little bit of a mess, I'm pretty sure I have a scanned version of something I did a previous semester, and I'll try to remember to post that up on Moodle. If I forget, just remind me, okay? I know it's a bit of a mess here. So we have some different bacilli, and we've talked about all of these in some degree, right? For example, Bacillus anthracis has a really distinctive look under a microscope. It kind of looks like spaghetti. Chances of people seeing that are pretty low. So other than that, you're getting just this normal kind of rod-shaped organism. But they do have different metabolic abilities. For example, the first two on there form endospores. Okay, so you can look for endospores. Clostridium tends to be anaerobic, and Bacillus tends to be aerobic. So those kind of things can help. So let me just write some notes on that. I'm just going to slide the screen over here. If I can remember how I did that, that's the problem. Okay, here we go. There we go. Okay. Where'd my pen go? Let's see here. What I will do is we can write in purple. So Bacillus. Endospore. So there are other metabolic tests, by the way, that can be done. But again, I'm not going to go into 100 different tests and have you guys memorize all these things. I'm just going to basically try to talk about what we've actually discussed already. So those are two things that we've discussed, right? Do they use oxygen? Do they form endospores? And actually just did a gram stain in one of my other classes, and we were looking at a Bacillus species, and there it was. You can see the endospores actually from the gram stain without any extra staining. It was just kind of obvious that you could see these little white specks in the corners of the cells, and I'll show you a picture of what that looks like in a couple of minutes. Okay, and Clostridium is anaerobic. And also an endospore former. Listeria is aerobic. I guess I wasn't going to write that, but one thing about Listeria is it's motile, right? So it has flagella and it can move around. So that's another test being done, and no spores. All right, I'm just looking to see if I have any other notes here. So sometimes people might look at glycocalyx, right? So they're looking for capsules or slime layers. Sometimes that's relevant with the Streptococcus, and there are special stains for them. They're not necessarily usually routine kind of things done for diagnosis. They're more or less, these are the kinds of things that scientists might do in terms of, okay, we have this new strain, let's characterize it and see why it's being so nasty. Oh, it has a slime layer, and that's why the immune system is having a hard time dealing with it or something like that. But like I said, not usually part of the diagnostic process. Usually we're looking for quicker tests. You know, the gram stain is pretty easy to do, and a lot of these metabolic tests are pretty common. Okay, so that's it for the gram positives. Hopefully everyone kind of got that. Like I said, I'll put up hopefully a little bit of a cleaner version on Moodle, and I'll go to the gram negatives in a second. Question?
Should we be technical, like let's say Catalase negative, or could we just say no problem?
Depends on what if this is an exam question, it depends on what it asks, right? Yeah. You know, it might say, what does a catalase test show, right? So in that case, you probably want to say bubbles, right? Yeah. Just kind of like I think I mentioned last day, if it asks you what a gram stain looks like, don't just say purple or pink, right? You know, say what the cell actually looks like. Do you see a Staphylococcus, a cluster of spheres, right? You know, because that's kind of a part of the observation, right? Is the cell shape and how they're arranged together. Okay, so gram negatives. So hopefully everybody got that. I'll just give you another quick. Oh, I'm trying to write on the screen. There we go. So there it is, all in one shot. I could probably post that on Moodle too. I think there's a way for me to save that as well. But anyway, gram negative. So like I said, they were going to be over here, but this tends to happen when I'm writing on the screen. I start writing bigger and bigger, and it gets a little bit weird. Okay, so gram negatives. So we have also three different kind of possibilities in terms of some of the cell shapes we're talking about, right? So we had 1, 2, 3. Okay, so Diplococcus, Bacillus, Vibrio. I'll draw some pictures of those. The Diplococcus. So some people talk about them looking like little bums, and they kind of do, right? Bacillus, of course, is a rod shape. You know, you wouldn't think it'd be that hard to draw this, but just this screen doesn't do what I want it to do. And then Vibrio are bent rods. So there we go. Okay, so Diplococcus. In terms of medically relevant organisms, there are a few, but 99-point-something% of the time, it's always Neisseria. And there's two Neisseria, gonorrhea and meningitis. So one is a sexually transmitted infection. The other is an infection that happens of the central nervous system, right? So depending on where you got the sample from, did you get it from the genitals or did you get it from a spinal tap? That's usually it's usually a quick and easy diagnosis. Okay, so let's write those down. We had, okay. Here's where I always got to double check the spelling for Neisseria. You know, it's always I before E, right? But not this time. Sorry, I'm just trying to figure out how I can just move this over here just a little bit. Got a couple of notes about those. So Neisseria gonorrhea, of course. What was I going to say about it? This is a sexually transmitted infection, and this is infection of, like I said, this the brain or spinal tissue, right? So usually that's it. That's the diagnosis, right? Like I said, you got the gram stain from that particular body part, and both of these are relatively treatable by antibiotics. Hopefully it's not too late in the disease progression kind of thing, and relatively straightforward. There are other tests that we haven't talked about, like for example, Neisseria gonorrhea is maltose positive. That means it eats the sugar maltose, and I think meningitis does not. And so sometimes those tests are done as a follow-up just to confirm the actual diagnosis. Okay, so the bacilli, here is where it gets extra complicated. Sorry, I'm just going to check the chat here. Okay, oh, come on. There we go. This over here. Okay, again, we have, unfortunately, we have many, okay, for Bacillus. And a bunch of organisms we've actually talked about already. So we talked about plague. So plague is Yersinia. And I'll be honest, I have no idea how to diagnose plague. I've never worked with it. Very few labs get a chance to do so, if you want to call it a chance. It's one of those things that you probably don't want to have to work with, right? But these other ones I do want to add some notes on. We also had E. coli. What else do we have here? Salmonella. Pseudomonas. So gram-negative rod, by the way, is a diagnosis for almost anything. There are like 100 species that can cause infections in humans. These are not necessarily the most common ones. These are pretty common. We could talk about something Klebsiella. We could talk about, there's a whole bunch of them, right? There's a handful that cause various foodborne illnesses and whatnot. Like I said, I'm really just trying to focus on certain organisms in this class because we can't cover everything. But just know there are a huge variety of gram-negative rods out there, okay? And that's where usually you're going into these further tests to distinguish what they are. And so I'll give you an idea of what we're looking for some of these, except for Yersinia, because I have no idea. Probably could look it up, though. Okay, so E. coli, what was I going to say about E. coli? So E. coli is motile. Lactase positive, I guess I didn't talk about lactase. It means it can consume lactose, but it's pink on McConkey agar. Maybe I can put that. Pink on, I have no space to put this. Pink on McConkey agar. I thought there was something else I was going to say about E. coli. There are special media. I think I showed you that that media, those plates where the E. coli turns out kind of a really interesting metallic green color. That's a pretty common method for looking at E. coli. And there are antigen tests looking at the different strains and whatnot, and a whole host of things, because when you start to get E. coli, there are different things going on in terms of there are some pathogenic strains, there are some sort of strains that are not normally pathogenic, but if they get into the wrong place, they can be. So E. coli kind of leads to, in some cases, a whole battery of other tests, some metabolic, some are antigenic, some are DNA tests, kind of depends on the whole scenario. Somebody has a blood infection versus just diarrhea. If it's just diarrhea, take your fluids and that's kind of the end of it. They're not necessarily going to, look at the organism and treat it, because usually you're looking at, 2 to 3 days kind of max for most people with E. coli diarrhea, right? So that kind of really depends on the context. But those are a couple of things we talked about. Motile, so they have flagella, lactase positive, and I mean, there's other tests as well. They're tryptophan-A positive, which means they have an enzyme called tryptophan-A. But again, we're not going to get into every single test you can think of. Okay, Salmonella. What was I going to say about Salmonella? Oh, one other note about E. coli. It's also catalase negative. So Salmonella is catalase positive. And it's yellow on MacConkey agar. It's also motile. So if you remember the MacConkey agar, it had a dye in it that when the organisms digest lactose, if they make an acidic byproduct, they turn yellow. That's for Salmonella. That's really kind of the bottom line. And there's a lot of tests where we want to know if it's E. coli or Salmonella. So something you may or may not know is if you go to any grocery store at all, and buy some ground beef, you will find E. coli in it. It is impossible to get rid of it. E. coli is found in fecal matter. So some of it gets in there probably from the slaughtering process of the animals, from human handling. But it's not found in very large amounts. But it's relatively easy to culture. You can find it in there. And that's okay. And that's part of the meat inspection process. They culture it and find out how much E. coli is found in that meat. And if it's below a certain threshold, then it's all good. And it's considered to be safe. Now, obviously, you still want to cook it, right? But if they find Salmonella, that's actually a different story. If they find Salmonella in there, it's like, no, this sample is contaminated, even if they find a very, very small amount. So this is one of these things. You know, they take the meat, literally take a small amount of it, they stir it up in some saline solution and spread that on a plate. And you can do that on MacConkey agar, and you can look for the presence of colonies. You find those little yellow colonies, it's Salmonella, and then therefore this is a bad batch, right? So very quick and easy test to do. It's something that I used to do with students in a second year microbiology course that we used to offer. And like I said, pretty easy lab to do, can be done in a couple of hours. Okay, so the last one is Pseudomonas. And Pseudomonas, we've kind of, I think I've mentioned it kind of twice. It's sort of, unfortunately, sort of gets pushed off to one of the last lectures of the course. But it's a gram-negative organism. And one thing about Pseudomonas is it's extremely opportunistic in that it rarely kind of infects you if you're healthy, but it's very good at when you're not causing some sort of infection. So it can cause all sorts of things. It can cause, it's famous for causing infections on people who are burns victims. So they've had a burn and it forms a biofilm on the burn, and it can be kind of a greenish color. It can give you pneumonia, it can give you ear infections, eye infections, like you name it. And so it's just very good at doing that kind of thing. And so we'll talk about Pseudomonas a little bit later on. Pseudomonas is lactase negative, meaning it cannot digest lactose. So we can put that down. What else is it going to say about Pseudomonas? It's catalase positive. I'm not going to say anything else about Pseudomonas at the moment. One quick way to find out if something is Pseudomonas too is it tends to be a lot more resistant to drugs. So you can put on selective media containing certain antibiotics that will easily kill E. coli and Salmonella, but not kill Pseudomonas. I think I referred to that before as Pseudomonas isolation agar. It's actually a formula you can just buy off the shelf, and you just put your culture on there, and it usually kills everything but Pseudomonas. It's kind of a little bit nasty that way, unfortunately. Okay, so one more, Vibrio. I'm just checking my notes, see if there's anything else I was going to look at here. And Vibrio, I'm going to just try to cram it in up there. And there's one Vibrio that, there's a few Vibrios out there, but the one that I'm going to mention once or twice later on this semester is the one that causes cholera. So it's called Vibrio cholera. Vibrio So that's kind of it for my flow chart, right? Trying to just take the ones that were in those tables I gave you and talk a little bit about them and some things to know about them in general. Vibrio cholera causes cholera, which is not a concern in Canada. As far as I know, there's never been a cholera outbreak in Canada, but I'm not sure, maybe going back 150 years or something like that. But it's very significant in many parts of the world. Cholera can be found in the environment in Asia, so it's pretty common for people in India to be part of, to get cholera from drinking water. And there's outbreaks that happen here or there, right? I can't think of what year that was. A number of years ago, could be 10 years ago now, there was a massive earthquake in Haiti. And of course, when you have an earthquake, you have a failure in public sanitation and whatnot. And just that it was at the wrong time for cholera to arrive to the country. And hundreds of thousands of people got sick with it. It was quite the disaster there. Okay, any questions? All right, good. Want to move on? Just check the chat. I think we're still all the same stuff going on. Okay, so there's my reminder to, of course, do this in lecture, and we did. Okay, so hopefully we'll get back on track here. So what I want to do is just show you the samples that I had swabbed and talk a little bit about the gram stains. I know I had done this very quickly at the end of last day. So this was somebody's hand. I think I told you before, it's super, super common for me to find Staphylococcus on almost all the samples that I swabbed, because Staphylococcus epidermidis is found on our skin. So pretty much everything that I've swabbed, I'm swabbing an actual hand or a doorknob or a keyboard, computer mouse, bank machine, they're all going to have Staphylococcus on it. So usually there's no surprise there. And this colony here is not yellow. So I know it's probably Staphylococcus epidermidis. There are a few other staphs out there. They're just not as abundant or kind of universal, right? There's one called Staphylococcus hominis that's associated with body odor, but it's not necessarily found on your hands. Associated with more moist parts of the body, like armpits, where you might find body odor, for example. Here's the keyboard, and same thing, right? So Staphylococcus, probably Staphylococcus epidermidis again. You could do further tests, like you could do the Catalase Test, and you could do the Coagulase Test and those kind of things to probably confirm it. But probably no need there. All right, here's the hand, and in this case here, I was going for that kind of it's kind of hard to tell on the screen, but it's sort of it's not as well defined. The edge is a little bit more. I'm not going to say fuzzy, because fuzzy sort of implies a fungus, but it's kind of pleomorphic, and it's a little more blob-like. And the reason why I wanted to go for that one is because I was suspecting Bacillus. Bacillus subtilis is often found in that kind of colony, and that's actually found as a very common soil organism, Bacillus subtilis, and we culture it sometimes in the lab for one of my other classes. So you can see, in fact, that this is a Bacillus, and so it's a gram-positive Bacillus. And if you look carefully, in the ends of some of the rods, there's some discoloration, so it's a little more white. And so that's actually evidence that these things are forming endospores. And see this once in a while, it's kind of cool, kind of exciting to always see that and look at it under a microscope. What else did I have here? So in this case, you can see in this case, I was picking on a different colony here, right? So notice it's not quite as light colored. And just in terms of the texture and whatnot, I was suspecting that that particular colony was E. coli. And in this case, I was actually right. So, or I'm assuming it's E. coli. It's the gram stain fits the profile of E. coli. But like I said, gram-negative rod could be like 1,000 different organisms. So this is from some keychain that was picked up. I can't remember whose keychain it was. I think it was a student's keychain at the time. It looks like that. You read the name there. Aisha or something like that, maybe? I can't remember. I think I was telling you about this last time, that I had these big Petri dishes that I bought. And so my colleague, up until 3 years ago, had an iPhone 4S, right? Like that's like from the Stone Age. But the cool thing about his iPhone is it had a really nice leather cover. And the leather cover had this nice texture to it. And when he showed me that phone, I was so excited because anything with a little bit of texture to it has a chance to capture bacteria. And so I said, can I borrow your phone for a minute? So I borrowed his phone, and look at that. You got this beautiful imprint, right? And you can even see the button on it and everything. And he was kind of excited to see the picture. He's like, this is good news, right? Anyway, I was just like, oh, maybe you should clean your phone once in a while, right? But anyway, so it's like you can't get away from Staphylococcus, right? You know, picture the colony. This here was a bit of mold. I wasn't gonna sample that, but this is probably, I can't remember the actual petri dish now, but all of those really look the same, and they're Staphylococcus. So like I said, usually after gram stain, in many cases, you've narrowed it down quite significantly, and in some cases, you're probably 99% sure what you have. Okay, so back to our bacteria, right? We've got the gram negatives, pink, gram positives, purple, and like I said, this is a very old test, more than like 150 years old now, to characterize bacteria. We're still using it because it's very, very useful. And like I said, some pretty easy diagnosis from some of these things. So back to our cell wall structures, right? So way back in topic, what was that? Topic 3, we were talking about bacteria, introduced gram negatives and gram positives, and I had mentioned that this kind of covers most of the bacteria out there. But in biology, there's always exceptions, and one of those exceptions are these guys here, the Mycobacteria. So the Mycobacteria, the most important one is the one called Mycobacterium tuberculosis, and these do not gram stain. So it turns out they're called Mycobacteria because they have these Mycolic Acids in their cell wall. And this is a cell wall picture from the textbook. And you can see it's kind of a little bit different, right? In some ways, it starts to look off like a gram positive. It actually has a reasonably, it's not a super thin layer of peptidoglycan. This picture is not showing that. It's actually thicker than it looks here. But then outside of the peptidoglycan, there is some other stuff. And the most notable are these mycolic acids, and they're very waxy. And waxy, like I've heard somebody say, it's kind of like crayon wax. So you can imagine if I was trying to, color some crayons, I put some crystal violet or something on there, it just washes off. It doesn't get in, right? And so if you try to gram stain these things, the stains just sort of wash off, and you don't really see much apparently. Plus, they're not gram positive or gram negative. They're something else. They're Mycobacterium. So we actually have a different stain for these, and it goes by a couple of names. Most people I know call it the acid-fast stain, and it's also called Zeal-Nielsen staining, which I think are two scientists that developed the stain. And so this particular stain has to do with the staining process. So the bacteria themselves are not acidic or fast. Okay, they're not speedy, anything like that. There is an acid used in the process, and fast means holding, right? So if I told you to stay fast, it might mean hold still, right? It's a term, one of those weird English flukes where we have a word that has multiple meanings, which is like half the English words out there, unfortunately. So what they're doing is to stain these things, you're using concentrated chemicals, acid, heat. You're trying to get the stain through that waxy layer, right? And so I'll show you how this process works here. So the acid is used to decolorize, and the fastness means secure, right? So that's what it means. So let's just talk about this procedure for a minute, right? So first you throw on this primary stain, which is called a carbonyl fuchsia, right? So what's fuchsia? What color is that? Pinkish, but like a hot pink, right? Like, I don't know if I'd say your pencil case is quite a fuchsia, but sort of, maybe? Yeah, something like that, right? So that's what carbonyl fuchsia is. It's just a fuchsia color, right? So this is the heat, and the heat melts the wax a little bit, and the stain is going to get in there, right? So that's the whole idea. Then you have a decolorization agent So this is a strong acid alcohol, and in many cases, this acid is enough to wash away the stain. But in the case of the acid-fast Mycobacterium cell, the wax is cooled off, and it's locked in the stain. So the acid-fast cells are going to be pink, and everything else is going to be uncolorized at this stage. And so then they throw in a counter stain, right? So the counter stain, remember, kind of just covers everything else. But the fuchsia is a very intense color. And so what happens is that this counter stain, the methylene blue, it's kind of a light color. It just colors everything else. But the fuchsia is such a bold color, it stands out, and you don't see the blue on the fuchsia cells. So that's how the procedure works, right? Like I said, there's other procedures for staining things. I think the textbook has at least one more procedure, but these are kind of the common ones. And this is the classical way for the diagnosis of tuberculosis, by the way. It's not, and by classical, I mean it's not really used so much anymore other than by scientists, because we have, there's a lot newer and better and more reliable methods, but it's important to know a little bit about it. So here, I thought I had some pictures here. There's a picture, and maybe you can see the pink, which is B. So these are the acid-fast bacteria, and then I'm guessing that's some Staphylococci. I don't know who did that exact picture there. I think I have another one here. This is the one from the textbook. This one here is from a, I believe it's from a sputum sample. Anyone know what sputum is? Yeah, it's something you cough up, right? So people with tuberculosis, that's kind of one of the classical symptoms, is a chronic cough. And that was what they used to say, okay, collect some of that in a cup, and then they would gram stain it, right? And so that's what you're looking at here. So if you look carefully, I'm not sure, sometimes in some of these stains, you see some of the ghostly human cells, but I don't think I can see that on that particular slide. There's another one. So these are some of the human cells that were coughed up right here. Usually they don't end up being so intensely blue, but you can see the little rods in there representing the tuberculosis. So much more in tuberculosis way sort of at the end of the course. Kind of mentioned here and there. Like I said, it's kind of a special organism for a number of reasons, and we're going to talk about it in a lot of detail in topic 14. So as I mentioned, there are other tests you can do. You can stain to look for capsules or slime layers. This is a particular stain called the negative stain, and the whole idea here is it kind of just stains the environment, but where the capsule or slime layer is, just doesn't, the stain doesn't collect there very well, and so you can see it looks like sort of these little ghostly shells around the bacteria here. And believe it or not, I think actually if I look at the textbook, this is Streptococcus. So they should be little round-shaped cells, but sometimes Streptococcus, kind of the cocci are almost stubby rods in some cases. And again, that's the kind of thing that a technician would know from experience. There are special stains to look at flagella. You cannot see flagella using a regular gram stain. They're just way too skinny. And so some of these special flagellar stains actually, they clump and build it up and make them look thicker. And that's how they're actually seen. And I've never done a flagellar stain. I think we have some slides that have pre-prepared specimens. There are other motility tests that are typically done, but just thought I'd show you this picture. It's kind of cool. And there are special stains for endospores. So I showed you the Bacillus. I got lucky, I could see them there. But there are special stains that actually show the contrast a lot better, and you can see there's one right there. Okay, so we were kind of talking about microscopy last day, right? And I was talking about better ways to improve your image, right? So you can magnify it by using a bigger lens, and that improves the magnification. It's kind of like think about a camera. You've got the guy with the little camera, and then you've got the guy with the big photo lens, right? You're going to get a better picture when you have a bigger lens, or at least you're going to zoom in more. You can improve resolution. Resolution is sharpness by having a better quality lens. So something that's not plastic, but you're using glass or quartz or some better material like that. and something that's manufactured with higher fidelity and all that. And the third thing we talked about was staining, and staining improves contrast. So that's kind of showing the edge between things, right? What I want to do now is just talk a little bit about fluorescence microscopy. And fluorescence microscopy was something that, you know, when I was a student, people were just starting to invest in it, right? So it's really come a long way, and it's really cool. This is showing a fluorescence microscope, and you can see sometimes they're using lasers and things like that. So they tend to be a bit more expensive. So a good quality compound microscope is about $8,000 to $10,000 for a good quality one. A good quality fluorescence microscope may be $25,000, right? So we're starting to increase on the price level, but we're not getting to unreasonable. So meaning that hospitals now will often have, you know, in their diagnostic lab, they'll have 1 or 2 fluorescence microscopes, right? And so not a big deal to, you know, particularly when you look at the advantages. I want to show you some of the advantages. So take a look at that, right? This is a fluorescent gram stain, right? So before I was, you know, kind of maybe squinting a little bit, and you know, and sometimes you do gram stain, you know, depending on, particularly when students do them, you know, it's like, is that kind of purplish? Is that kind of pink? You know, sometimes there's a bit of that goes on. Fluorescence, you get really, you know, the contrast is amazing. So this is really just kind of a cool type of staining is really what fluorescence is in many ways, right? You're staining something, but now the stain actually glows, right? So very, very cool. Sometimes you get some amazing, cell biologists are really into fluorescence nowadays because you can get some really amazing detailed images. You can see there's some epithelial cells. You can see that the nucleus is stained one fluorescent color blue, and the microtubules are stained the green, and you get some really incredible pictures. There's Giardia, like I said, these pictures, they just pop out, okay? And you get some amazing details. And there's some spindle fibers and chromosomes. They're you know, ready to go. This is anaphase, right? So some very cool pictures. There's a worm, I don't know. I was just looking for some fluorescence pictures, and I thought that was kind of interesting. There's another one. I don't know what they're doing, but they made a rainbow cell. Lots of interesting things being done with fluorescence. So I told you a basic fluorescence microscope is maybe $25,000. It might be used in a diagnostic lab. A lot of researchers are spending way more on these things because part of it has to do with the software and sometimes has to do with the hardware. So one of the big differences that I'm starting to see a lot in the research is people using something called confocal fluorescence microscopy. So normal fluorescence microscopy, sometimes you have this problem. If you're looking at a eukaryotic cell, it's pretty big, right? So picture like, you know, if I have, well, let's say this here, right? So I'm reading this here, but all of you in the background look blurry. And as soon as I focus on you, the writing looks blurry. And this is the same problem with a microscope. You can only focus on one thing at a time. You have this big three-dimensional object, big in the micrometer range. The microscope is so sensitive, it can only focus on parts of the specimen at a time. So these confocal microscopes use lasers. And the lasers, what they will do is they will focus in right here. And then it can focus in right here. And then focus in right here. And you can build up a much sharper image. And so now you're looking at maybe $100,000, right? Plus, you know, $50,000 for the hardware, $50,000 just for the software that can interpret it. But some very cool stuff done with confocal microscopes. This one here, this is some sort of a blood-brain barrier in a zebrafish. Some very, very fascinating pictures. This one here is one of my favorites, up close and personal. I like the name of it. You can see the nucleus right there in the front. Those yellow things are mitochondria, and the purple is some cytoskeletal structures. So very, very cool. There's another one. Some of these are from these, I think this is Nikon that does this. They have a photo micrography contest because they sell microscopes too, besides cameras. And so the best pictures pop up every year. There's another one, some sort of skin cell. So this is getting like I said, hospitals are investing in these because they're very, very handy for diagnosis. This is a blood smear, a fluorescence blood smear looking for Plasmodium, which causes malaria. And it's very similar to doing a traditional blood smear. You take a little bit of blood, it gets smeared on a slide, there's a staining process, and then you have a trained technician looking at the slide to diagnose the malaria. But, you know, trained technician, right? That's the key thing. They got to know not only how to do the process, but they also have to know what to look for, right? In terms of, you know, what's going on with that blood smear. Fluorescence, like anybody here can tell you, you know, assuming you're not colorblind, there's yellow blobs on there and they look just like the Plasmodium parasite, right? Like, you know, there's no question. It's so easy. And so this technology, you know, is, like I said, the diagnosis is much more certain. Yeah, it costs more, but it's saving everyone time and money, and you know, you're not getting any false diagnosis. You're not getting a case where the technician has to call their supervisor and say, "What do you think? I think we need another blood sample," right? Like, we don't want to mess with that. So for you know, a bit more money, we can do some amazing things. There's some. I guess I meant to show this one here before. I was mentioning that confocal, right? I was mentioning you can focus here and focus here and here, and you can actually build up three-dimensional shapes, and that's actually what this particular person had done. They're building a three-dimensional shape of, what do we have here, Giardia and Cryptosporidium all in one sample. I would hate to have a double infection of that. So here's some more diagnosis, right? So you can see, you know, you can imagine your typical clinical specimen could have all sorts of other gunk in there, right? So this specimen on the left here, you got mucus and debris. I could do gram stain on that. And sometimes there's just so much there. All you see is this blob of purple, and you don't know, is it that's because there's a bunch of gram positives, or is it because we have just all that mucus has trapped the stain, right? Done under fluorescence, and boom, you can see those bacilli very, very easily. So fluorescence has a lot of huge advantages, and usually when I go to conferences now, they're selling all sorts of new fluorescent stains, and they sound expensive, but like I said, you can imagine the time that is saved with these kind of things, and having that certainty that you do have what you're looking for. So traditional fluorescence confocal, I think I mentioned that already. Okay, so a lot of fluorescence is specific stains, right? And it's worth mentioning that there's one technique that is also really starting to become super important both in diagnostic labs and in hospitals. And this is called immunofluorescence. So we're going to talk about the immune system. That's kind of later on in the semester, topic 13, but we are going to talk a little bit about immune methods in this topic today. And one of these immune methods is immunofluorescence. So one of the important parts of the immune system are antibodies. Have you guys talked about that in anatomy and physiology yet, antibodies? Not yet, you'll get there. So antibodies are little proteins. They kind of look like Y-shaped proteins, right? So they're the part of the immune system that your body generates when it has an infection, and then it helps you to recognize the next infection, right? So if you've had a vaccine, or if you had chickenpox as a child, now you have cells that are producing these antibody proteins, and these antibody proteins will recognize that infection if you ever get exposed again, and they're going to deal with it. So much more than that in topic 13. But think about it. We have this little protein, and this little protein is super specific for something. So it could be the chickenpox virus. It could be the spike protein on COVID-19. It could be a surface glycoprotein on the Plasmodium parasite. It could be some sort of carbohydrate on Salmonella. So you've got a very specific protein that binds a very specific thing. So this is great. It's kind of like spycraft technology, right? Where you are identifying that one thing in a billion, you know, like the facial recognition software. Except for now you have a protein that is doing that same thing with some sort of infectious particle or something else, right? Because it doesn't have to be infectious particle. This could be, you know, an allergy, maybe allergic to peanuts or something. And so the antibody is the protein, the antigen is the foreign particle. So we can take these And we can make them in animals, so we can make them in mice or rabbits or goats or something like that. And we can harvest these proteins, have them in a test tube, and we can get a chemist to come along, and a chemist can make that fluorescent, right? So here's kind of a little picture showing how that might work. There's the little antibodies. The little green things are a fluorescent dye that has been added on by some sort of chemist. And so now we have these little very specific antibodies that are going to target something, and they glow. So this is great. So if you take a look here, it looks like these ones are targeting Neisseria gonorrhea. And then under the microscope, they're all going to congregate, and you're going to see the gonorrhea, right? So like I said, you've got very specific way to bind something. And there are catalogs like this thick now of these companies. They have thousands and thousands of antibodies in their catalogs, and researchers and diagnostic labs use them all the time because somebody else has gone through the trouble of making this for you. Like I said, usually a mouse or a rabbit is pretty common, and having them chemically modified so that they fluoresce. Back in the old days, it used to be radioactivity. Actually, not that long ago, I was using some radioactivity, but we don't want to use radioactivity because it's radioactive and has a whole bunch of other issues. But fluorescence, it glows, and this is kind of awesome. There's a picture of somebody actually detecting gonorrhea there from that source. Here's another one. I'm not sure. This is from it's from a different textbook, not ours, and they were showing somebody who had some fluorescent labels for plaque, right? So you can see the rod shape of the Yersinia cells there. But same idea. Okay, I'll get back to that in a second. Okay, so immunofluorescence, super cool, super specific. So I know I had in the notes, I had this little table, right? Because I'm going to talk about electron microscopy for a minute and kind of compare and contrast those things. And but this is just kind of a summary of some of the things that we've talked about, right? So you've got you're using light to illuminate your specimen. The light could be from a candle, it could be from the sun, it could be from a light bulb, it could be a laser, and that's making, showing the specimen. And you have some sort of lens. The best quality lenses are made of quartz. Glass is not quite as good. Plastic is not, as good. Although there are some new polymers and whatnot out there that people are formulating, and a lot of them are used in actual eyewear, like reading glasses nowadays. Specimens are in color. They can be dead or alive, depending on what you're trying to do with your research. Some of the techniques we talked about, oil immersion. Did we talk about oil immersion? Maybe we didn't. Don't worry about oil immersion. Talk about it in my other class. Staining and fluorescence are kind of some of the two massive techniques that have really helped us to do things in diagnostic labs. It's relatively easy and cheap technology, and the magnification, up to about 1,000 times. With some of the advanced fluorescence stuff, we're actually really breaking that barrier to 2,000 times. But that's really pushing the limits of what can be done with actual visible light. So I want to talk about for a few minutes, just a brief amount of time, about electron microscopes. If you don't get all this, I'm coming back to this slide in a minute, okay? But what I want to do is talk about electron microscopes and what's going on there, okay? So I don't have a Kahoot for you, sorry. I started it and I never finished it. So I will post it for you to do on your own time, but I never got finished putting it together. Sorry about that. It's been a busy week. So light microscope on the left, electron microscope on the right. So what's the whole idea here? So tiny bit of physics for you. If you look at a wavelength and what a wavelength of light can do, it can only have a certain theoretical resolution. So that theoretical resolution is maybe something like 200 nanometers, which means you can't see anything smaller than 200 nanometers. In fact, in most cases, you can't see anything smaller than 1,000 nanometers with a light microscope. But electrons can have waves, and those electron waves theoretically can see things much smaller, and that's the whole idea behind electron microscope. So if you look here at your light microscope, you've got a light source, you've got a lens, you've got a specimen, and then you're looking at the specimen. And there's our specimen, our little mosquito eyeball. A light microscope, same idea, except for you don't have light, you have an electron beam, right? So an electron beam, what are the lenses made of? The lenses are not glass or quartz. The lenses are actually a magnetic field. So you can focus that electron beam, and you're not going to stick your eyeball in there to look at it because, of course, you're shooting the thing with a high energy electron beam, not good for your eyeball, but you're going to have some sort of photographic paper, or most modern microscopes are using some sort of digital detector, and the image ends up on a computer screen, right? So I want to talk a little bit about this electron microscopy. I've actually done a little bit myself. This is the instrument that I use at the University of Guelph. And this one was purchased in the 1980s, right? These things are expensive. So I think this cost $100,000 in like 1989 or something like that, right? So in today's money, that's like about $1 million or something, I'm not sure. But either way, it was expensive and it still works. So you're not going to buy a new one. What you're actually looking at here, this is the microscope here. Over here on the left, the big tall thing is the gun. That thing on the right is sort of, I guess you could call it an old computer. And they're actually not shown in this picture is the actual computer attached to this. And this thing on the right, it's actually just the knobs are used for focusing, but the computer part isn't used anymore. Here's some of the images that I took. And so this particular technique is called scanning electron microscopy. And you can see it, you kind of get a surface image. So the idea behind scanning electron microscopy is the electrons kind of hit the surface and bounce off, and then they're read by a detector. Notice that most of the pictures are grayscale. Electrons don't have colors. There are not blue electrons or orange electrons or purple electrons. You can add the color after. And so you get really amazing kind of just pop-up pictures. And so a lot of textbooks use these because they look very, very nice. Here is our textbook. I think maybe that's actually the first edition. And you can see some Treponema. That's the organism that causes syphilis. On the left, you can see Giardia. So you get a lot of really cool images. And a lot of these are not used in hospitals unless they're research hospitals because you're looking at very expensive equipment. And mostly you're looking at scientific research. But in many cases, you know, when we had the new coronavirus, people wanted to see what it looked like right away. And so, you know, it is an important part of scientific discovery and kind of our knowledge. Some more pictures for you. I love the name of this one, right? This was the person who took it and published it. They called it, you know, a face only the mother could love, right? Mosquitoes, right? I don't like mosquitoes. If I could just wipe one thing off the planet, you know, and just be happy of it, right? It would be mosquitoes. So I want to talk a little bit about how we can actually do this, just give you an idea of how it works. So there's a specimen. If you take a look, that's actually a person with a mosquito on the end of some tweezers. And the specimen gets put on a little platform, and that little platform gets put into this instrument here. This instrument is called a Sputter Coater. I don't know how the Sputter Coater works. The one I used, you stick it in, you press start, you walk away for a minute. And but what it does, somehow it spits little gold particles onto your specimen. So why gold? Because gold is a heavy metal, it has tons of electrons. So it's kind of like staining your specimen in electron microscopy fashion, right? Something with lots of electrons is going to show up a lot better. So there's the sample. If you kind of look, it's a little shiny. That's because it has a very, very thin gold layer on it. Then it goes to the microscope. The microscope itself has this big fat door and these big handles. And what you do is you have to crank it shut. And then when you turn it on, there's actually a vacuum pump, and it pumps all the air up. So air, of course, has electrons. So you're trying to remove any background noise that's going to be on there. And then basically you stick it in, and you can focus in on your specimen. And so I was trained on how to use this. I think it took about a day to train me how to prepare and focus and use the specimens. And then there we go. There's a bug. I didn't take the bug pictures. I kind of wish I had. I was looking at bacteria, but here's some bugs. You can zoom in. You can see we can zoom in on the hair on the bug. And in fact, we were zooming in, and you can see some detail on the hair on the bug. And one more, I think. And in fact, you can zoom in so much, you can actually see the individual proteins on the hair on the bug. So this is just really incredible stuff. We're seeing very, very tiny things. Question?
So in SEM, is it necessary to stain these specimens?
It's not necessary, but good luck seeing much if you don't. Because most organic things are made of carbon, nitrogen, oxygen, which are, sort of lower elements. And there are some techniques that don't use it, but it's usually more for research, right? Or sometimes people might have a stain that's maybe, let's say, only stains the DNA or something like that. And so so everything else is going to look ghostly. And then that one thing you're looking for might come out nice and sharp if you have a good stain. Yeah, good question. Here's some pictures, horrifying pictures of parasites. Every time I look at these, I'm like, if you're making a science fiction movie, you don't need to make anything up. Just make a giant tapeworm or a hookworm or something like that. Like, look at this thing, the hookworm. If I saw this in a dark alleyway coming at me, I would be terrified. So notice the three-dimensional images, right? So this is the scanning electron microscopy. There's one other I want to show you that is used a lot as well, because sometimes we want to see into things, right? By the way, one of the key things to know about electron microscopy for this course and is that you can see viruses. Right now, we can see small enough, we can actually see virus particles. If you take a look here, this is actually the coronavirus that causes COVID-19. And there's the original image. And as I mentioned, we can add color. And notice, this image is great, the one on the left. It's actually very good. But throwing in the color, it's just like, it just pops out at me, right? And of course, everyone loves color, right? Unless you're my brother-in-law, who is colorblind, and he doesn't get color. It was kind of interesting when he married my sister. She loves flowers and colors and painting and all these kind of things, and he kind of just kept his mouth shut, when she would say things like, "Aren't these flowers lovely?" "Yes, dear." Because to him, they were just like various shades of brown, apparently. Anyway, another story on that. There's another one. This is some HIV viron particles that are budding away from an infected immune cell. And again, the color kind of just allows it to pop out, and you can see the virus particles a lot better. There's some human chromosomes, just kind of cool. Thought I'd show you that. Don't need an electron microscope to see human chromosomes, but this one's just kind of cool. And there's Ebola. A bunch of Ebola viruses breaking out of this cell, and it's kind of Ebola is a very spaghetti-like virus, and you can see them there. And there's a bacteriophage, so you can see some detail on that virus there. So very cool. And then just for fun, right? People get artsy, salt and pepper. Anyway. Okay, let's talk about one other kind of electron microscope, and this is the transmission electron microscope. So transmission means that the electrons actually go through the specimen. So this actually destroys the specimen, by the way, if you keep the microscope on long enough, but you can get pictures of what's going on inside a cell or a specimen, and I'll show you some pictures here in a moment. These are also usually stained. Like I was saying, sometimes you're staining something in particular. I did a little bit of this myself as well, but this was the kind of thing where they're like, how often are you going to use this? Okay, once? All right, we're not really going to train you. It took me a week to prepare my specimens with the guy kind of sitting over my shoulder the whole time, and I couldn't actually touch the actual microscope because it just wasn't worth their time to train me. It takes too long if you're only using it once in a while. I don't know if I have yeah, there's my picture. Didn't even turn out very well. I wasn't very happy about that. But you can see that these spots here are probably ribosomes. You can see the two membranes. This is E. coli. And that's about all I could see. So it wasn't very useful in my case. But in many cases, people are getting some great images. This is from one of my textbooks I had on my shelf. And you can see the nucleoid region. You can actually see a little bit of detail on the cell surface and whatnot. And I'll show you some better pictures here. I think I have some. Here's a eukaryotic cell. You can see the nucleus very well in the center there, the giant circle. And this is, I think, a muscle cell, and it's loaded with mitochondria. So very, very cool, right? We can get some internal details. There's a malaria parasite. So some color. You can add color to these as well to make them pop out and look a lot nicer. and that's usually what people do for certain publications or textbooks or things like that. There's some viruses, and when we had, so 2 pandemic viruses right there for you all in one slide. So 2 for 1 deal, right? So 2009, H1N1 influenza pandemic. This hit the news relatively early on in the pandemic, and this was over all sorts of publications, this exact image. Although it came in different colors because the CDC, I think, had the picture and they had several colors. There's one of the coronavirus. So I think I told you corona means crown or something like that. And so the person who named it in the 1960s thought it kind of looked like a crown with little jewels on it. And so that's where coronavirus comes from. And you can see they've colored it so you can see those spike proteins, very well on that particular micrograph. Okay, so that's kind of it for electron microscopy. Just want to show you the difference. There's a ground stain, scanning electron microscopy, and there's a transmission electron microscope. So all of them have different uses. Electron microscopes are more for on the research end. Hospitals aren't going to spend, like I said, a scanning electron microscope, you could probably get a cheap one for a couple $100,000. Transmission electron microscope, you could probably get a cheap one for $500,000. But usually you're spending a lot more because you want a decent quality, unless you're buying used, then you can maybe get a cheap one on, I don't know if they sell them on eBay. I did find one once, by the way. I don't know what made me think of it. I was on eBay once and I put in electron microscope. And there was actually one on there for $25,000, a used one. I don't know who's buying them off of eBay, but I thought that was interesting. So back to this slide here. So just kind of comparing these two things, right? Get an idea of what's going on here, right? So illumination now, we're using electrons. The lenses, we're using a magnetic field, okay? So, just the hardware in general is a lot more expensive. In this case here, the specimen is always dead. You're drying out the specimen, you're coating it with heavy metals. Sometimes those heavy metals are toxic. You're not looking at anything living. So that's a disadvantage for scientists who sometimes want to observe living processes, right? There's several techniques. Most of the techniques do fall into the 2 categories of scanning or transmission, SEM or TEM microscopy. It's expensive. The training is quite extensive in some cases, and so that means you have to hire somebody who has that training, and it's not like it's necessarily done everywhere, right? We're talking about usually people with grad school experience. And what kind of magnification? So SEM up to 250,000 times, TEM up to 1 million. I think actually there's some newer microscopes that can do 2 million. It's really incredible. And the key thing is we can see viruses now. So we can actually understand a little bit about virus biology, or at least enhance our understanding using electron microscopes. Okay, see a couple of people writing madly. Let me see where we are with time. I'll just give you another moment. Probably finish up here right away because I know there's some other things to talk about that are probably just worth it to wait until next lecture. Okay, so moving on. If you do have a big budget, you can buy this thing. This is, what is it, the Hitachi H3000. This is apparently the most powerful transmission electron microscope. And if you take a look at this, it is huge, right? Those are railings. So like you get a ladder to get up to that first level, and then there's a massive electron gun. I think there's a picture here. I thought there's a picture of a guy working in it. So some of these transmission electron microscopes, I used to work with a fella, and he was able to do this one technique using a transmission electron microscope, and he estimated there were 5 or 6 people in the world that had been able to do what he had done, right? So these are pretty highly trained individuals. And like I said, we're looking at mostly research only. But if you've got several $1 million, you're looking for a cool pastime, here you go. So I wanted to spend the last 2 minutes just talking about one newer type of technique, and this is just for fun only, will not be on the midterm. This is something called probe microscopy or scanning probe microscopy. There's a whole bunch of techniques. This was invented in the 1980s. And so now picture if you want to look at a specimen and you're blind, right? So this is kind of, that's the whole idea. So you can imagine a blind person, right? They might go along and, you know, they're like feeling something, right? And, you know, if it's, okay, this is, I can feel it. It's long and skinny and it's got some bark on it. That's a tree, right? You feel this next thing and it's furry and it's licking my fingers. That's a dog, right? So this is a bit more sophisticated in that it literally scans. It has a probe that kind of crawls up and down the specimen. And as it goes along, it feels the contours. So this type of technique is so sensitive, it can actually detect individual atoms. It's very, very cool. And there's a whole bunch of techniques, and they all have different acronyms. The one that's used the most often in biology is called AFM, or atomic force microscopy. You can see in this case here, this particular molecule was ran through, and you can actually see the shape. You can almost see the atoms in that. Usually we're talking about if you want to see atoms, you need to see big things and hard things like metal surfaces. Turns out with biological specimens, they're a little bit more squishy. So it's a little harder to get down to the atom level. But we can see some pretty cool things. And the color is added afterwards. So you can see here's some samples of E. coli. You can see the flagella. Sometimes they get a little warped. Like I said, these specimens tend to be a little more squishy because they're biological. And that's kind of normal. You can see here's some people looking at viruses using atomic force microscopy. You can see the herpes virus on the left. You can actually make out the individual capsid proteins. So this is research only, kind of getting cool, won't be on the midterm. I just thought I'd share with you because it's some kind of really cool cutting edge technology. This was invented in the 1980s. And up until about 15 years ago, if you wanted one of these microscopes, you had to build it yourself. You can actually, in the last 10 to 15 years, buy them now. So there's been a lot more people using them in various places. One last image I want to show you. I think this one's just super cool. Looking at DNA, and you probably know that DNA is a helix. It's this double helix, and you can see that on the left, so you're looking at is a model. On the right is the actual image. So they've actually been able to see and prove that DNA has that helical structure. Now notice all the lines. Like I said, this biological specimen is kind of squishy. It's not perfect. It kind of moves and wobbles around as they take it, but you can clearly see the helical structure. Okay. That's where I'm going to finish today. Next day, we'll talk about serological stuff, and we're going to talk about genetic DNA methods and all those kind of things. And we'll get into topic 9 as well.